Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요. And hello all. Welcome and thank you, SynBYSS people, for joining us, the 108th seminar, and I call it on MIT Day. Happy New Year. I hope all of you enjoyed great holidays and started a hopeful and exciting 2024. Since December 16, I have stayed at home without any trips for almost 4 weeks. It was great to have dinners and lunches with local friends and family, and it was relaxing after more than 150 days of trip last year. In fact, I'll embark on my first trip of 2024 this weekend to visit Singapore, where our rising star speaker today is located, although I am visiting National University of Singapore rather than Nanyang Technological University, where today's speaker currently located. So what a coincidence, I would say. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Hadley Sikes. She is a professor of the Chemical Engineering Department at MIT. She also leads labs at MIT as well as in Singapore as part of Singapore MIT Alliance for Research and Technology called SMART. I may need to call today a Singapore Day because of some common thing today. Importantly, she served as ACS, American Chemical Society, Division of Biochemical Technology Program Chair very recently, organizing the last year's ACS Biot Conference program. That's a very, very important job organizing and make people gather together. Hadley, thank you so much for your service to the community and excellent research and dedication to education. One thing I want to mention, she's the graduate program officer. That means she's a very important person to accept all the PhD program people and the master program people at MIT Chemical Engineering program. And I know how busy she is right now because this is probably the busiest season of the year today, but still she joined us today. Thank you so much again. The virtual podium is all yours now. Thank you.
Thank you. Thank you so much for inviting me. And I am delighted to be able to introduce today's young seminar speaker and topic. And I'm going to do that a little briefly, referring to a couple of examples from my own work, why I think kinetic modeling, kinetic analysis is so interesting and important to synthetic biology. So our research projects, the theme is using molecular biotechnology to diagnose disease and to treat it more effectively. And as Professor Moon mentioned, we have our labs spread across MIT and Singapore. And we work together from the discovery ends of things, understanding the systems we work in at a fundamental level, to clinical testing. A lot of our clinical testing goes on in the Singapore lab. And the examples, a couple of brief examples. One of the tools that we've worked to develop is a paper test to diagnose disease. Unfortunately, during the pandemic, we all became all too familiar with doing the COVID home tests. And one of our goals has to make them faster, taking only a couple of seconds instead of 15 minutes, more sustainable, made totally out of paper without plastic in the end, and also more affordable and more stable so that they can be used all over the globe in countries where there's no cold chain storage, and these kind of diagnostic tools are really needed. And so the SynBio there is, instead of antibodies, we engineered a protein from a thermophilic organism to function like an antibody. So we changed the binding face to make it bind whatever target protein we're looking for in a bodily fluid. And another example of a kind of diagnostic test, not for diagnosing disease, but for once a patient has already been diagnosed, for example, cancer, one of the big challenges is to pick out which therapy is going to be effective for that person's individual disease, rather than going through a trial and error process where you try a first-line therapeutic, there are awful side effects, it may work or it may not. And so in our research, what we're doing is we're asking the question of what can we measure in a patient biopsy to be able to know whether that person's individual disease is going to respond to therapies or not. And the particular class of therapies is oxidant-inducing therapies. And so there's a bunch of oxidant-inducing therapies that are approved by the FDA, but their mechanisms of action are not always clear. And so we've used synthetic biology approaches to discover more about the mechanisms of the oxidant-inducing therapies. Which oxidant are they inducing? And is that a bystander kind of effect, or is that causing the death of the cells? And so we've made tools for that. And then also we've come up with tests that one can do of a patient biopsy to decide whether that therapy should be treated with that, whether that patient's disease should be treated with that particular oxidant-inducing therapy, yes or no. So that's a really high-level overview of what we're trying to do and how it interacts, or how synthetic biology is involved. All of our projects start with the first technical step is kinetic analysis, kinetic modeling. The very first step is clinical needs assessment, going to doctors, going to clinicians, asking them about what are their unmet needs, what tools would they like to have that they don't currently have, and what are the design criteria? What are the tools that we build? What are their characteristics need to be? And so in the paper test example, how that intersects with kinetic modeling is the doctors told us they wanted tests that were faster within an office visit, and they wanted them to be more sensitive. And so we went back to the lab and we said, okay, sensitivity and speed, we know we have equations for that. We're going to need to understand, as a patient sample flows through this test, there's a binding reaction rate, there's convection rates, there's diffusive rates. And so this guides our choice of our synthetic biology efforts. And so in this particular case, when we did this analysis, it told us that we can get faster assay speed and better sensitivity if the new protein part is smaller than an antibody and integrated into the system at a concentration that takes advantage of the smaller size. So in this residence time and how much of the analyte are we losing, how much is just flowing right by, it told us we wanted to be down on this orange curve, which is not where rapid tests currently are. So we wanted to be on this orange curve. We wanted something small. The binding rate would be really fast, and we could pack it in there. And so that guided us. And the lateral flow tests that we've all used during the pandemic are up on these curves, where during the time that the sample spends in contact with the line, a lot of it just flows through. And that's a lost opportunity for greater sensitivity. So kinetic analysis told us that. And then similarly with the tumor biopsies and selecting oxidant-inducing therapies, there are reaction networks in cells and different compartments of cells, the cytosol, mitochondria, and so on. And thanks to omic studies, we know which proteins are involved in the network. We have a whole bunch of rate constants for reactions of particular oxidants with proteins. We can model those reaction networks, and we can do sensitivity analysis to ask which protein in the network is going to be most sensitive to changes, to oxidation reactions in the range that has physiological relevance. And so we did that, and it told us we should focus our SynBio, our synthetic biology efforts on this particular protein, peroxyredoxin, the isoform in the mitochondria, peroxyredoxin 3 in particular. And so my goal is just to say that kinetic modeling, kinetic analysis, in addition to being fascinating, it's really useful. And I'm excited to hear what our young speaker today has to say in his interesting story.
Thank you so much. I entire your talk, I keep smiling because you remind me of around this time of the year, but almost 18 years ago, I took a qualifying exam at MIT Chemical Engineering Department, and then one of the exam was the written exam, and then at the time we have the oral exam. Basically, I need to understand the paper assigned to me, but that paper is very similar to what you just described, the drug delivery, but using kinetic model and the transport phenomena analysis. I should kind of understand and convey things to the professors. And then, oh my goodness, this what a coincidence. I absolutely enjoy what you just said. Thank you so much. And this is why chemical engineering people excel in the synthetic biology, because we have the tool to apply to analyze the complex system, I believe. Absolutely brilliant. Thank you. Okay, so now the main speaker of today with a longer introduction. Dr. Luan Lu is a postdoctoral researcher at Nanyang Technological University. He first studied as a material engineer. Later, he changed his field to biophysics when performing his PhD project in enzyme kinetic modeling at Nanyang Technological University, Singapore, where I visited last year to give a seminar. Also, Hadley also has lab in the Singapore. That's another common thing I just mentioned earlier. And interestingly, basically Dr. Sikes, talk about kinetic modeling, but his field is also related to kinetic modeling at current institute. His research interests are physical modeling and data analysis to understand biological processes, including protein synthesis by ribosome and energy generation by F1 ATPase. And I believe that two big macromolecule is probably one of the most important molecule in biology, I believe. And I especially thank him for his time today, because it is midnight, actually after midnight right now in Singapore. So Luan, thank you so much again, and please take it away. It's all yours now. Thank you.
Yeah. So I would like to thank Dr. Moon for spending your time organizing the event, and Professor Sikes for your interest in my presentation. And let me start my presentation by share screen. Can you see my screen?
Yes, but can you make the slide mode?
Yeah. Okay, so it's good, right?
Yeah, wonderful. Thank you.
Yeah. Okay. So thank you, everyone, and let me present my research, which focuses on the kinetic modeling of 7DS ribosome, which is shown on the left, and the F1 ATPase shown on the right. First and foremost, I would like to thank the help and the support of the people I have worked with throughout the years. They are Dr. Rudy Marcus, Dr. Su Haibin, Dr. Sandor von Kankapso, Dr. Chu Kai Cheng, and other members of the Dr. Su group. I also thank the financial support of Iron Fusion Fellowship. We all know that cells are the basic building block of life. For their survival and growth, cells have to intake the food, break it down to produce energies, and assemble building blocks into functional proteins. Therefore, cellular metabolism requires two basic things: The first thing is functional proteins, and the proteins are synthesized by ribosome, which decode the information from the messenger RNA into the chains of a polypeptide that can later fall into functional proteins. And the second thing is energy stored in terms of ATP molecules. And these molecules are synthesized by an enzyme known as F1 ATPase. As you can see in the cartoon, the ATP is synthesized within the F1 component. And in this talk, I will first focus on the ribosome, followed by the F1 ATPase. So inside the living cells, the ribosome have to recognize the correct substrate. And this correct substrate is called the cognate. Oh yeah, I forgot. Let me change to the pointer to laser. Easier, yeah. So this cognate has the correct base pairing between the codon from the messenger RNA and the anticodon from the transfer tRNAs. However, inside the cells, there are incorrect substrates, which has structures very similar to the cognate with just one base pair difference. And this kind of incorrect substrate is called decognate, explaining just their very similar structures. In terms of kinetics, what people have measured in the test tube is that the rate of GTP hydrolysis for the correct substrate, like for different type of correct substrate here, as you can see at 1 millimolar, the speed is around 30 to 50. And then as the magnesium concentration increases, the speed will gradually increases and reaches plateau. In contrast, for the case of incorrect substrate, the decognate, the speed here, as you can see, is significantly slower. However, when the magnesium concentration increases, the rate of increase here is significantly faster compared to the correct substrate. When you combine these two data together, and when you plot the speed of processing the correct substrate versus the accuracy, defined as the speed ratio between the correct versus the incorrect one, for different type of species, different type of substrate, and you can see here, when you increase the magnesium concentration from low, you can see the accuracy is high, but the speed is low. So when the magnesium concentration increases, when you follow the line here, you see the accuracy gradually reduces while the speed increases. Therefore, it leads to a kind of trade-off. If you want to increase the speed, you have to stratify the accuracies or vice versa. Slow down the speed and get higher accuracy. This kind of speed and accuracy trade-off is observed for the whole reaction pathway of ribosome shown on the left here, which comprises of initial selection and the proofreading. And the figure on the right here shows you how the speed versus accuracy for the initial selection and the whole reaction pathway, as seen over here, where the trade-off really dominant for both stages and the whole pathway. And this kind of trade-off can be explained by a simple model proposed by Hadley. In this model, the ribosome will rapidly equilibrate with the substrate, followed by a fast hydrolysis to enter a kind of activated state, enable it to either forming a peptide bond or slowly reject the substrate and restart the whole cycle again. Therefore, this kind of rejection will enable it's called proofreading, enable the ribosome a second chance to improve the accuracy. However, because you reject, the more you reject, there will be less species available to form the peptide bond, lead to less product and then slower the speed, leading to a trade-off. If you want more accuracy, you need to sacrifice the speed. The limitation of this model is that it's not really explicitly capture the competition between the right and wrong substrate, especially in the living cell, with the relevant biological recursor. And because so far the trade-off is observed based on the experiment in vitro. How about what happened in the living cells? So we will already do some kind of mutation experiment to observe this. So what they did is they mutate the cells to make the ribosome more error prone. It means that it's less accurate, or make it more accurate, hyper accurate, and then observe the growth rate and compare it with the wild type. So the result here shows you that the mutant cells, they have growth rates lower than the wild type. So based on this result, people propose a hypothesis that actually, is it really possible for the cell to the accuracy to maximize the growth rate? If this is true, this means that other than the trade-off between the protein synthesis speed and accuracy, there should exist another type of positive correlation between speed and accuracy, where both the quantities simultaneously increase or reduce. Before dive into that, you need to understand how the ribosome and the growth rate are related to each other. And experimentally, people have found that the ribosomal mass fraction is linearly dependent on the growth rate, as indicated by this graph over here, where you see the x-axis is the growth rate and the y-axis is the ribosomal mass fraction, represented in terms of the ratio between the mass of the RNA over the mass of the total protein. And here is a series of data points connected by changing the nutrient qualities. So as you can see, when the nutrient quality improves, the growth rate increases, and the mass fraction of ribosome also increases in a linear manner. This kind of linear relationship can be explained by a simple model whereby there's kind of a supply and balance. There's kind of balance between the supply and demand of amino acids. In this model, the amino acids is produced by proteins that have to import the nutrient and produce and convert them to amino acids. The amino acids are then synthesized by the ribosome, which have to synthesize either ribosome or the proteins themselves. So basically, they have the ribosome and the proteins, and the graph here shows you how the growth rate can be changed by tuning the ribosome fraction for each nutrient condition, as indicated by different colors here. For one type of condition, you can see the growth rate can vary as the mass fraction of the ribosome, and they exist more or less like an optimal growth rate. So across different nutrient conditions, you can have optimal growth rate, and this optimal growth rate is more or less linear, and hence it can explain the experimental result. Other than the mass fraction of the ribosome, people also care about actual quantities such as the cell mass, and experimentally, the cell mass on average is linearly dependent on the growth rate. And another quantity of interest is the metabolic rate, how much energy consumption the cell requires for its growth. And experimentally, people found that under its active state, for the bacteria, it follows power law with the exponent of 2 for the metabolic rate scaling with the cell mass. So taking it all together, we found out that there's a kind of needs we need to address, where we need a model that can really capture how at the molecular level, how the cell can overcome the competition effect between the correct substrate versus incorrect substrate, where this one is much more in terms of numbers, and with different type of kinetics between the correct versus incorrect substrate, so that the speed and the accuracy of the ribosome will really influence the cell growth. And we will need to relate how different cellular properties such as the cell mass, the number of ribosome, the metabolic flux, and the growth rate are related to each other. So let me come to present my first key result, where I try to address this problem. So the left figure here basically show you the cycle of protein synthesis. And the approach we do is we try to simplify this circle into a simple one shown here. And this simple model is nothing but an extension of a few model. And the correspondence between the two figures are as follows. So you see this one, the rotating thing is the free ribosome waiting for the substrate to enter. And then once the substrate binds to the ribosome, if you flow through all these linking state, and then in this circle, this is captured by the effective arrow here, indicating the effective rate. The green one is for the correct substrate, the connect, and the red one is for the incorrect substrate, the deconnect. The thickness of the arrow indicates the magnitude of the rate constant. Once they go through all these states, they will reach this blinking one is the GTP activated state, followed by the hydrolysis to enter the proofreading. And then this kind of proofreading, the ribosome, they can either reject the substrate or forming a peptide bond. And both of the processes will enable the ribosome to enter the free state and restart the whole cycle again until it completely transfer the messenger RNA. So based on this simple model, we can really effectively write down the equation that capture the overall protein synthesis speed as well as the accuracy. And the expression actually nothing but can be based on the line. This FC and FNC here correspond to the amount or the fraction of correct and incorrect substrate inside the cell. And all of the K here are the elementary rate constant indicated in this figure, in this cartoon. And based on this model, combined with the experimental data on the magnesium concentration at different kinetics, what we found out is that what really matters are the 3 states of ribosome. The first one is the free ribosome, the next one, or at the ribosome at the proofreading RPR here, where the green one is for the correct substrate, the ribosome and the group reading associated with the correct substrate. And the red one is the ribosome and the group reading associated with the incorrect substrate, the NC. So what you can see is they are the function of magnesium concentration. So as around 1 millimolar, you can see the dominant is the green one here. And then when you increase the magnesium concentration from 1 to 3, you can see because of the increase in the association kinetic up between the ribosome and both the correct and incorrect substrate, the green one increases and the red one also increase, and therefore the free ribosome will be reduced. From 3 to 8 millimolar, you can see that a significant drop in the green one, and it is converted to the ribosome accumulating for associating with the wrong substrate. The accumulation of the wrong substrate here is because the ribosome significantly, because the kinetic binding between the ribosome and the wrong one significantly increase faster. It's a much faster rate, and together they have slower rejection at the group reading. And the accumulation of these wrong species, right, will affect the speed and accuracy as shown on the figure over here, where this axis is the effective protein synthesis speed, this axis is the magnesium concentration, and this axis is the accuracy of selection, defined as how the flux of forming peptide bond by the correct substrate over the incorrect substrate. So you can see this accumulation here will correspond to a regime here known as competition. So you can see for this one, for around like 3 to 8 millimolar, the protein synthesis speed will gradually reduce when the magnesium concentration increases. When you see on the blend of the speed versus the accuracy, this competition regime here, you can see the speed will reduce and simultaneously the accuracy also reduce. Other than this competition regime, there's from 1 to 3 millimolar, there's a kind of trade-off which I have presented earlier, where when you increase the speed, you have to, like this one, you need to sacrifice the accuracy. So effectively, from this blend point of view, you see that from 1 millimolar to 8 millimolar of magnesium concentration, there are kind of like optimal or fastest protein synthesis speed. At this point, acting as the crossover point between the two regimes. And based on this kind of relationship, we can extend it to study how the protein synthesis inside the living cell. And this is shown on the graph over here, where this is the speed, this is the accuracy on the x-axis, and this one is the KMF. This KMF and is nothing but a parameter expressed in this expression. So you see that for the triangle here, it will be the overall protein synthesis speed, and they are nothing but the function of K cell Cmax and the KMF, where the K cell Cmax is the open circles and the KMFs are the open square. And effectively, these two parameters, they can be correlated with the accuracy at different magnesium concentration, and hence overall capture how the overall protein synthesis speed will behave. Based on this symbol expression, we can readily capture them together with the amino acid flux balance to study the bacterial growth by having this kind of expression over here, where you can really see that how the maximum protein synthesis speed and the Km here are the function of can be considered as the function of accuracy. So the task to simulate and model this kind of model here is to, for each magnesium concentration, we just try to vary the UR here is the how much the cell will allocate to ribosome and the remaining to the proteins. And therefore we can optimize the growth rate. And then based on at different optimal growth rate, we can choose the fastest one. And the outcome for this kind of procedure is shown on this graph over here, where I plot the optimal growth rate as the function of accuracy. And this different accuracy will correspond to different magnesium concentration. So different curves over here with different colors correspond to different nutrient qualities. And so for each curve, you can see a small open circles. They correspond to the maximized growth rate for each accuracy. So for the K of cell, among almost these possible growth rate, they may properly just choose or pick the optimal one. So you can see that from poor to rich nutrient condition, or of the optimal accuracy, optimal growth rate, indicated by the solid circle, they gradually shifted from the high accuracy here to the low accuracy, while the growth rate gradually increases. So the result here suggests that when the neutron quality improve, the cell tend to sacrifice a bit of the accuracy to achieve higher growth rate. Even though the accuracy reduced from like around 30,000 to this range 3,000, the values of accuracy are still quite high enough, and the cell can tolerate the error max during the protein synthesis. And another outcome, the implication from this result is that we assume or we predict that the cell can really regulate and tune the magnesium concentration so that they can tune the accuracy to optimize the growth rate. And in reality, what people found out that it may be really possible, like in the case of antibiotic threat over here, where the antibiotics will attack the ribosomes. The cell can have some process to import more magnesium concentration to stabilize and protect the integrity of the ribosome structures. So you can see on this graph here, when the ribosome is having more extra magnesium concentration, you can see the number of hyperpolarized cells is smaller, and they have higher elongation rate or protein synthesis speed compared to the ones that are subject to antibiotics. Another scenario is under hyperosmotic stress. So under this scenario, there are the cell will produce some signaling molecule that will trigger the cell to import magnesium concentration and accumulate up inside the cell, so that the osmotic pressure difference between the cell inside the cell and the outside environment are not so significant, are not so different, so that they can survive. Another point I want to highlight is after we achieve the optimal growth rate indicated over here, we can see how they relate with other cellular properties. And here is the graph. The inset here will show you roughly how the growth rate and the ribosomal mass function relate to each other. And it's a linear relationship consistent with the one found in experiment. And what we found further is that there are some kind of a quadratic relationship between the growth rate and the number of the ribosome. So this is a square root. So when you square, it follows show that the number of ribosomes really quadratically dependent on the growth rate. This quadratic dependence is based on the linear relationship between the mass function of the ribosome and the growth rate, and together with the linear relationship observed between the cell mass and the growth rate. And another relationship we found is the quadratic relationship between Jm here, which is the neutron intake flux, which can more or less reflect the metabolic activity of the cells. And this is follow more or less quadratically on the cell mass, like shown on here, which is consistent with the experimental result. When you plot them in the log scale and compare with other experimental data, our result fit nicely and consistent with the report for the E. coli. And the slope of this log law is 1.96, which is very close to the square root of the value 2 here, suggesting that protein synthesis is really an important metabolic activity of the cell. So putting all together, what I have presented is a multi-scale model where at the molecular level, we capture how the speed and the accuracy of the protein synthesis look like and related to each other by extending a few model, where the protein or the overall speed can be expressed in terms of simple expression, which is also known as Michelin-Menten expression, where the maximal speed and the Michelin constant here actually can be related to the accuracy, and they can be modulated by the magnesium concentration. So when we plug this to a bacterial growth model, what we can find out is that the cell can really regulate. What we suggest is that the cell can really regulate the magnesium concentration so that they can tune the accuracy and the protein synthesis speed in order to maximize the growth rate for different nutrient conditions. And the outcome of this optimization process is that the linear relationship between the mass function of the ribosome and the growth rate, and the cell mass versus the growth rate. Based on this linear relationship, we can further show how the number of ribosomes will be quadratically dependent on the growth rate, and similarly, the nutrient intake flux, which is the indicator of the metabolic activities, also quadratically dependent on the cell mass. And this is the end for the ribosome part. Let me move on to the F1 ATPase part. So for the F1 ATPase, which is the highlighted, it is the rectangular box and this cartoon here, and which important ingredients are the gamma axis and the 3 beta blockers. So what experimentally, what people did is they will attach a kind of probe onto the gamma axis here and observe its rotation, like this kind of figures here, where you can see the revolution as a function of time. And inside this kind of trajectory, what you see is really a kind of long dells, then a rapid jump, and then long dells, and then rapid jump, and then so on and so forth. So based on this kind of trajectory, what people have found out is the type of chemical reaction really going on in these beta pockets, which is shown over here. So you can see that there are three chemical states. The first state is, we call it PI release del at around 0 degree. At this state, if you're waiting for the PI to release, once the PI release, it will trigger the gamma axis rotation by 40 degrees. So you can see this arrow here indicating the gamma axis already rotated, has been rotated by 40 degrees. At this binding del of 40 degrees is the empty pocket here of the empty red pocket here. You wait for the ATP to bind, and this binding of ATP will trigger another 80-degree rotation to the catalytic dells. And at this state, you can see throughout this process, the ATP in the blue one is more or less released. And then at this state here, the ATP in this green one is ready to be hydrolyzed, waiting for hydrolysis to occur. So conceptually, this whole three state will occur throughout 120-degree rotation. So you can see that for one revolution of 360 degrees, there will be three 120-degree transitions. And there are also some interesting properties about the kinetics of the F1 ATPase. The first one is that some rate constant, such as the binding of the ATP molecules, they are exponentially dependent on the rotation angle. So because this is the log scale, so you can see it's linearly related to the rotation angle. Similarly, for other rate constant like the PI release here, like this one, they are also more or less linear. So it's meaning that they are exponentially dependent on the angle for the chemical reaction rate. Another interesting property is the angular jump, which indicates the rotation speed of the enzyme, which is reflected by observing the rotation of the probe. Because what we really observe is the probe, but then it will indicate how fast is the rotation of the gamma axis of the enzyme. So when the probe is very huge, at the micrometer size, you can see it's approximately around constant. But then when people manage to improve the size, it reduced the size of the probe and increase the time resolution, what people found is actually the rotation or the jump, the angular jump of the enzyme actually varies with the rotation angle. So overall, what I have presented to you is what people found out is about the chemical kinetics of the enzyme with different state and different rate constant. And one side is, and the other side is the trajectory of how the angular jump vary with the rotation angle. But the question remaining is, how do we quantitatively connect the two here by addressing these three chemical states and their kinetics, and how this kind of kinetics will influence the rotation of the probe that are subject to the fluctuation of the viscous medium surrounding them. So with this kind of question we want to address, let me present to you our first main result. First, what we did is we tried to statistically see the distribution of the angles throughout the whole trajectory. So because the dells are the longest, so what you can see is throughout these peaks are the position within the dells. So you can see the gap between all of these peaks, they are not exactly 120 degrees, suggesting an even distribution. Based on this uneven one, because of this uneven one, what we did is we need to manually choose the transition that can consistently fall within 120 degrees plus minus 10 degrees that are consistent with the experimental result. And the inset here will roughly show you how fast the transition look like. So you can see the transition is more or less around 120 microseconds. And throughout this short transition time, there will be three chemical states I mentioned before. And the quantity we are interested in to calculating is the angular jump between the transition, which is approximated by the angular difference between the two data points divided by the time step. The time step here is just the camera time frame, of just 10 microseconds. And here will come our first best result. So based on the kinetic data and the three chemical state, what we have, we will connect them with the heat map of how the angular jump and the rotation angle are distributed. So you can see here that with this state, with the distribution, you can see a bunch of high probability state around here will be 0 and located around, mostly around 40 degrees to 50 degrees, and we will assign it to the binding tails. And from 70 degrees and beyond, we can safely assign them to the state number 3 here. And we have also come up with a theoretical model that can qualitatively capture the experimental result. The way we come up with the theoretical model is by simply assuming that the probe or the enzyme is confined within a harmonic potential subject to viscous fluctuation. And what we really want to care about is the probability distribution for each chemical state as a function of time. You see this term. So the factors affecting this evolution is, we call here the drift term, is how the bit will be tend to relaxes to its minimum potential. The diffusion here is because of the fluctuation with the surrounding medium. The minus term here indicating how fast the enzyme will leave the current state I. And this term indicating how fast the enzyme will enter from other state to this current state I. All of the rate constant here, forward and backward, are exponentially dependent on the X. And based on this histogram, this heat map, what I also want to highlight to you is that at some angle like 42 degrees and 66 degrees, you can see that mostly they are just one peak occurs in the distribution. However, for some angle in between, like around 60 degrees, there are kind of two peaks occur here, indicating the bimodal distribution. So you see this bimodal distribution suggests to you that at the same rotation angle, sometimes the enzyme can be in state number 2 or state number 3. The reason for this is because the chemical reactions are too fast, while the speed is much huge, and it will slowly respond to the chemical reaction. So even though the probe is at 60 degrees, the enzyme already at jump from state number 2 to state number 3, resulting in this kind of bimodal distribution. So, and then based on this distribution of the angular jump versus rotation angle, what we will also study the mean angular jump as the function of rotation angle, which are the scatter dot over here. So when you can see that when we use the 3 state, which is the dashed line, there's a good agreement from 0 to 40 and from 80 to 120 degrees. However, there's a big gap around 40 to 80 degrees. This kind of big gap, right? We postulate that it can be reconciled by having another extra meta state named 2 star. This extra state is shown over here, where the lifetime is very short, 10 microseconds, and located around 70 degrees. The physical meaning of this state is that they are triple occupied for both 3 blockers with ATP, ADP, and ATP. This lifetime is so short is that it because experimentally people found that without the ATP binding, the release of ADP takes very long, second to minutes. So our reasoning is that the occurrence of ATP in this red pocket really helps to induce the ADP release in this blue one. Thereby, the lifetime is significantly reduced to just 10 microseconds. Another interesting thing I want to present to you is this region number 3, where you can see the scatter points more or less follow a linear line. The slope of this line can indicate to you the viscoelastic relaxation time of the probe is around 50 microseconds. Another way for you to double check and provide the consistency to this result is by plotting the position correlation at the del. And then when you plot this and extract the relaxation time, this is around 40 microseconds, consistent with this one, suggesting that our results are consistent and having a coherent framework. So overall, what I have presented to you is based on the current knowledge of step number 2 and step number 3 of the F1 ATPase, what we focus on is the transition zone here, and we delve on to study the distribution of the angular jump at different rotation angle. And what we show is that sometimes it's just a modal distribution, but then sometimes they can be bipodal, indicating the fast kinetic of the enzyme and the slow response of the probe. Furthermore, when we quantitatively show how the mean angular jump and the rotation angle behave, we found out that there may be an extra state which is very short in between around 70 degrees that has the physical meaning that there's a kind of pocket coupling where the ATP binding in the red pocket can induce and transfer the energy of its binding to enhance and accelerate the release of ADP in this blue pocket. So that I make it very short and difficult to detect. And with this, I would like to end my presentation, and thank you very much.
Wonderful. Thank you so much. I mean, it's absolutely great talk. Professor Sikes, do you have any comment or question?
Yeah, definitely. Thank you. That was really interesting. I thought both topics were distinctive. You know, that kind of work you don't see it that often. And I particularly liked in your first story on slide 11 that visualization of the reaction mechanism. Yeah, your over there, your coarse grain 1, where you show the different rate constants, their magnitude with the different colors and the different line widths for the accurate and inaccurate one. I thought that was really, really effective. I'm going to steal that idea.
Yeah.
So a question after that comment was having the detailed quantitative information here for the system that you studied, which is E. coli, right? Have others done it with different kinds of microbes or with yeasts or humans or mice or just across different organisms? Do those magnitudes vary?
For what I know so far, people just focus on bacteria, so mostly E. coli.
Okay.
Other species I don't know.
On slide 15, I think you had E. coli. You had the colored dots. What are the gray dots? What are the gray ones?
So all these all these ones, they are different bacteria.
Okay.
Yeah.
So there's some variation on a log plot again across different bacteria.
Yes. But this is the metabolic rate. So and in this graph here, you can see it's a different paper correspond to, I think, no, I think they are still E. coli. I remember these are all E. coli. So far, the result, they mostly focus on E. coli. Only this one, they are different bacterial species. Yeah.
And do you have any more question or comment?
Yeah, I have going up a few, those were kind of technical questions, but going up a few levels with the beautiful detailed understanding you have of either of these two kind of systems, what kind of practical implications come to your mind? Now that we understand those systems in such great quantitative detail, does it enable new practical insights? And if you don't know yet, that's fine. I have to confess, my own PhD was on quantum mechanics. It involved Marcus theory. It was not related to anything I'm doing now. And I still, like 20 years later, don't know what the practical implications are. So not every research has to have a practical implication.
Yeah. So so I think one future research direction I would like to be involved with is, because what I've done is I relate the speed and accuracy together. So what I really want to see is when the ribosome transfer the messenger RNAs, what the probability of having the correct versus incorrect substitution for each codon. So when you have the message, then you can see that the sequence of polypeptide, there will be correct and incorrect amino acids, right? So how this kind of incorrect amino acids can fold and affect the protein folding. Because by understanding this, we can predict, we can like have some application like in the, like for example, the COVID vaccine, already like try to synthesize the proteins from the mRNA chains, right? So at this fundamental level, we really want to understand if there's kind of a misinformation incorporated during the transcript, during the translation, how it will affect the protein folding. Given that now we had AlphaFold, so in principle, we can calculate at what point of the codon, what's in the probability of having a wrong amino acid, and what type of amino acid is wrongly substituted. And based on that information, we can predict how it affects the final protein structures and eventually the function of the proteins. Yeah, I think that's the long-term vision that I think this work can be extended.
That's an interesting point. Let me jump in. Actually, the Hadley's question is like exactly my question. Because I'm the engineer, I'm already thinking about application, the practical application down the road. To me, I mean, so I kind of keep thinking about your remark about trade-off between speed and accuracy, and this is not surprising thing, because I mean, if you make things faster, there probably the less accuracy over there. But the example you gave that about the RNA vaccine thing makes sense, but at the same time, to me, I mean, if you model sort of increase the accuracy somehow during the practical application, I will buy it. However, if you somehow make the accuracy same, but at the same time make things faster, I'm not necessarily convinced, because the entire time scale of the transcription will be much faster than other process time, like the purification, etc. So I believe if your work eventually translated, there should be more for the accuracy increase rather than the speed increase, because that's probably the most important aspect, I believe. But I might be wrong, but then that's my comment about that example. But that's good example. Now I understand better. So Professor Sikes, do you have any other question, comment?
I don't think so. Thank you. You taught me something. I appreciate it.
Thank you so much. Yeah, so my question is answered, so I don't have any more question. Entire time I feel like I became again the graduate student, learning more about chemical engineering core principles. This kind of refresh my memories. That's the great thing to have, first seminar of this year. So thank you so much. I believe now, almost the 11 of my time, and that's close. So thank you so much for joining and staying today. And we will meet again on January 18, Thursday, the same time, the same Zoom link. We'll have Professor Jeff Bork at New York University, Rangoon Health, and Dr. Tetsu Hamimoto at Harvard University. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thanks. I will stop recording. Thank you. Just give me 1 second.